3 on biological classification. So in the last lecture, we studied about 5 kingdom classification that was proposed by R.H. Whittaker. So it was known as Whittaker's 5 kingdom classification, according to which all the organisms were divided into 5 kingdoms, majorly into 5 kingdoms that were Monera, that mainly includes prokaryotes. Then we have Protista, then we have fungi, then we have plantae and animalia. So monerans were prokaryotic cells, while the remaining ones were the eukaryotic cells. And based on the other criteria, these eukaryotic organisms were further divided into these 4 kingdoms, that is Protista, plantae, animalia and fungi. Now, although it was one of the well-established method of biological classification, still with the advancement in the technologies, with the advancement in our understanding about the cellular and genetic composition of the organisms, the classification system is also getting improved day by day. So after the 5 kingdom classification, there were certain limitations to the 5 kingdom classification that I would first like to highlight. So a major problem was that it lacks the distinction between the archaea and bacteria. So the monerans according to the 5 kingdom classification include both bacteria and archaebacterium. So however, there are certain differences between the archaebacteria and bacteria in their cell wall composition, in their method of reproduction, etc. So that we will understand in the later lectures. But the major problem with this 5 kingdom classification was that it lacks the distinction between the archaebacteria and bacteria. So this was one of the limitations in 5 kingdom classification. Also, the kingdom Protista, that includes protozoa, algae, etc., is very diverse. Although the organisms in the kingdom Protista are all eukaryotic cells, but the organisms that are present or that were included in the kingdom Protista were very diverse. So it is difficult to call them as one group. And later on, when the classification system was further refined, these organisms were put into different other kingdoms. So the kingdom Protista was very diverse, either based on nutrition also we can say, because in the last lecture we understand that the organisms that belong to kingdom Protista were both autotrophic as well as heterotrophic. So it was a very diverse kind of organisms were included in the same kingdom. So this is also one of the limitations. Also, in addition to this, the boundaries between the kingdom Protista, plantae and fungi are ill-defined. That is, although there were certain features based on which the kingdom plantae was separated from the kingdom fungi and kingdom Protista, but the boundaries are not very well defined. One of the characteristics that was used was the cell wall composition, but beside that, there were no other such boundaries that were used to define the kingdom in a more appropriate way. So there are certain examples. For example, we can see the brown algae were probably not closely related to the plants, even though the 5 kingdom system places them in the plantae. So now when we have genetic information about the organisms, now when we know the genetic composition of the organisms, when we can understand the genetic similarities between the different organisms, so now we have the understanding that the brown algae is nowhere or not at all closely related to the plants. But the 5 kingdom classification placed this in the kingdom plantae. So there were certain limitations. Few of the organisms were put into the, we can say, were inappropriately placed into the inappropriate kingdom. So the refinement with the advancement in the technologies had led to their better placement in a appropriate kingdom. So after this 5 kingdom classification, so all the lectures will focused on the 5 kingdom classification only, but just for the knowledge, for the sake of knowledge, whatever advancement that has taken place in this biological classification system, I will be telling you about the 6 kingdom classification and 8 kingdom classification as well. So this 6 kingdom classification was put forward by Carl Weiss. So 5 kingdom classification was put forward by R.H. Whittaker. Before that, we learn about the 3 kingdom classification and 2 kingdom classification. So this is a 6 kingdom classification, which was proposed by Carl Weiss, who was an American microbiologist, and he proposed this 6 kingdom classification in 1990. So this classification system was a departure from the traditional 5 kingdom classification, where all the organisms were classified into the 5 kingdom based on their cellular structure, mode of nutrition, or on the basis of their cell wall structure, etc. Now, from this 6 kingdom classification, newer technologies, or we can say the sequencing, was used to analyze the similarities and differences between the different organisms. So to compare the different organisms, Carl Weiss has basically used the comparison of the ribosomal RNA sequences, which is also known as the rRNA sequences. So based on the comparison of rRNA sequences, this classification system reflects the evolutionary relationship between the organism, and based on that phylogenetic or evolutionary relationship, the organism were placed into the different kingdoms. So more closely related organisms, which were having the more which are phylogenetically more related, which have same evolutionary history, were placed into the similar kingdom. So basically the work is based on the molecular phylogeny. So molecular phylogeny is is interpreting the evolutionary history based on their molecular sequences. So based on the rRNA sequences, it leads to the recognition of 3 domains of life, which is the bacteria, archaebacteria, and eukarya. So basically, it is also known as the 3-domain system of classification, which is indeed a 6-kingdom classification. So basically, there were 3 domains that were defined. The one is bacteria. So we can now see that archaebacteria or archae are put into the different kingdom from 6-kingdom classification forward. So basically, these defined three domains of life. Which one is? So the first one is bacteria. The second one is archae, and third one is the eukarya. So bacteria, archaebacteria, and eukarya. So the classification was done into 3 domains, and then these domains can further be divided into kingdoms. So basically, we can say that it is a 6-kingdom classification where the organisms were divided into bacteria, archaebacteria, protista, plantae, fungi, and animalia. So this system is reflects more about the evolutionary history. It considered more about the phylogenetic relationship among the different organisms to place them more accurately into the appropriate kingdom. So unlike the 5-kingdom classification that was proposed by R.H. Whittaker, where both the monoreans and both bacteria and archaebacteria were put into the same kingdom, which is the monera, the 6-kingdom classification put them into 3 domains, which is the bacteria, archaebacteria, and eukarya. And this domain was further divided into the kingdoms, that is bacteria, archaebacteria, protista, plantae, fungi, and animalia. So basically, the system aimed to reflect the evolutionary relationship. So this is very important, that before that, much focus was not given to evolutionary relationship, and therefore there were some inappropriately organisms were put inappropriately into some different kingdoms. But from now, the focus was more shifted towards the evolutionary relationship. It was based more on the molecular phylogeny based on the rRNA sequencing, that is the ribosomal RNA sequencing. So based on which we can identify the molecular phylogeny of the organism, its evolutionary history, and therefore based on that, Carl Weiss put them into 3 domains of the life, as I mentioned, bacteria, archaebacteria, and eukarya, and which can further be said into the classification of organisms into 6 kingdoms, that is bacteria, archaebacteria, protista, plantae, fungi, and animalia. So this was about the 6-kingdom classification. The next and the more detailed classification is of Cavalier-Smith. So Smith basically proposed the 8-kingdom classification system. So till now we were studying the 5 kingdom or 6 kingdom, but the Smith proposed the 8 kingdom classification system. And here he has used the term empire and kingdoms. So in 6 kingdom classification, 2 terms were used, domains and kingdom. But in this 8 kingdom classification system, two terms that were used were empires and the kingdoms. So basically, in 6 kingdom classification, we have 3 domains and 6 kingdoms, but in this we have 2 empires and 8 kingdoms. So in this, all the organisms were divided into 2 empires. The first empire is empire bacteria. The second is empire eukaryota. And these empires were further divided into the kingdoms. So empire bacteria, as we can see here in this diagram, consists of 2 kingdoms, which is the eubacteria, that is true bacteria, and archaeobacteria, which is archaebacteria or the primitive bacteria. So the categorization was done into 2 empires and 8 kingdoms based on their cellular structure and genetic organization. So in this 8 kingdom classification system, the focus was given to both the cellular structure and the genetic organization of the organism, just like the 6 kingdom classification. But with the advancement, we have understood better about the sequencing methods and better about the other genetic tools. Therefore, this provides a more broad and more classified system of classification of biological organisms. So basically, the empire bacteria, as I mentioned, were divided into 2 groups, eubacteria and archaebacteria. And the empire eukaryota were divided into the 6 kingdoms, which is archezoa, protozoa, kingdom plantae, kingdom chromista, kingdom fungi, and kingdom animalia. So in this, we can see that in the earlier classification, there was a term which was used as kingdom protista. But from now onwards, in 8 kingdom classification, we do not have any kingdom protista. One problem with the kingdom protista was that it was very diverse. So due to its diversity, it was difficult to place all the organisms in the same kingdom. So therefore, because of the diverse nature of the organisms that were placed into the kingdom protista, they were further placed into the different other kingdoms which were defined in 8 classification system, that is archezoa, protozoa, and chromista. Remaining plantae, animalia, and fungi were same as previous one, but other new kingdoms were added into this 8 kingdom classification, which were archezoa, protozoa, and chromista. So the archezoa kingdom comprises primitive unicellular eukaryotes. Just like archaebacteria, which were primitive bacteria, archezoa represents the primitive unicellular eukaryotes like Giardia, which was characterized by the 70s ribosome and the absence of Golgi apparatus, mitochondria, chloroplast, and peroxisome. So the organisms that were lacking, so the eukaryotic organisms that were lacking the Golgi apparatus, mitochondria, chloroplast, and peroxisome, for example, Giardia, were placed into the kingdom Archezoa. The second kingdom that was added as chromista. It includes primarily the photosynthetic organisms with chloroplast situated in the rough endoplasmic reticulum, but not the cytoplasmic matrix. So there is a difference. Here we can have the confusion about the presence of the chloroplast. So the chloroplast is a photosynthetic pigment even in the kingdom Plantae. But what is the difference here is that in this, the chloroplast is situated in the ARIA, that is the rough endoplasmic reticulum, but not in the cytoplasmic matrix. Unlike the plants, where the chloroplast is present in the cytoplasmic matrix. So this was the difference, and all the photosynthetic unicellular eukaryotes, so the photosynthetic eukaryotes were placed into the kingdom Chromista. All the photosynthetic protista that were earlier grouped together with the non-photosynthetic organisms in the kingdom Protista were now put into the different kingdom, which is the kingdom Chromista. So the Chromista kingdom includes primarily the photosynthetic organisms, or we can say the photosynthetic protista of the earlier kingdom classification, that were having the chloroplast, which is situated in the rough endoplasmic reticulum and not the cytoplasmic matrix. So this is a line of difference between the kingdom Chromista and kingdom Plantae, where the chloroplast is present in the cytoplasmic matrix in plants, but in the rough endoplasmic reticulum in case of kingdom Chromista. So based on this classification, the organisms like diatoms, brown algae, which was earlier, as I mentioned, were placed into the kingdom Plantae, were now placed into the kingdom Chromista. So now, with the addition of the new kingdom, which is the kingdom Chromista, there is a better classification of the system into the better groups. So diatoms, brown algae, cryptomoniads, and oomycetes, those were photosynthetic protista, were now placed into the kingdom Chromista. And therefore, based on this, the boundaries of the kingdom Plantae, fungi, animalia, and protozoa kingdoms have been refined to enhance the clarity and differentiation between them. So with the advancement, we can see that we are able to set the boundaries in a more appropriate way, in a more refined way. Therefore, by setting the refined boundaries, we are able to put more similar organisms into a similar type of kingdom, into the same kingdom. So this was the 8 kingdom classification, which was proposed by Cavalier Smith. And this is not the end of classification system, basically, because with the advancement, as the techniques are getting, as we are learning more about the organisms, as we are advancing in the technology, in the field of technology, we are able to better understand the genetic composition in the molecular phylogeny. And with this increase in the understanding, with this increase in the knowledge about the phylogenetic relationship between the organisms and their genetic makeup, we are able to better classify the organisms into the more appropriate manner. So therefore, we cannot say that this is the best classification system, but yes, still now it is, we can say, but maybe in future we might have a much better classification system of organisms that classify the organisms into maybe many more different kingdoms based on their genetic composition or cellular structure or many different characteristics. So this was about the different classification system that we need to know. Starting from the 2 kingdom classification, where organisms were only classified into kingdom Plantae and Animalia. Then we have 3 kingdom classification, where the organisms were classified into fungi, Plantae, and Animalia. Then we have 5 kingdom classification, where prokaryotes were given a place by introducing a kingdom Monera. Then we have after 5 kingdom, we have 6 kingdom classification, and now we have 8 kingdom classification as well. So this was about different type of classification system that has been proposed in biological sciences. So after this, we will be starting or focusing with the first kingdom, which is the kingdom Monera of 5 kingdom classification. But before proceeding to the kingdom Monera, I would first like to give you a brief introduction about the eukaryotic and prokaryotic cells. Like what exactly are eukaryotic cells and prokaryotic cells? First, this is one of the major points based on which the kingdom Monera is defined. And we know that all the prokaryotes are placed into the kingdom Monera. But what exactly is prokaryotic cell? So basically, we know that cell is defined by a membrane. So the cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nuclei, like the cells which does not have membrane-bound nucleus, are prokaryotic. So the eukaryotic cells and prokaryotic cells are differentiated based on whether they have a cell nuclei which is defined by the membrane or not. So the eukaryotic cells are the one in which the nuclei is surrounded by the membrane, while the prokaryotic cells are the one that lacks the membrane-bound nucleus. So this is the figure where we can see that this is a plant cell, which is itself a kind of eukaryotic cell. This is an animal cell, which is also a eukaryotic cell. Here we can see that the central part represents the nucleus. And this nucleus is surrounded by a membrane, which is a nuclear membrane, which we can clearly see in this, right? So this is a nucleus, which is being surrounded by a membrane, which is a nuclear membrane. But in prokaryotic cell, on the other hand, we will see it's a naked DNA. So naked DNA means a DNA which is lying freely into the cytoplasm, not surrounded by any membrane. But in eukaryotic cell, we can clearly see that it is a membrane-bound nucleus. So a nuclear membrane is present, that is, a membrane surrounds the nucleus. So this is one of the major distinctions between a eukaryotic cell and a prokaryotic cell. In both prokaryotic and eukaryotic cell, a semi-fluid matrix called cytoplasm occupies the volume of the cell. So cytoplasm is present in both eukaryotic and prokaryotic cells, and most of the activities that takes place, all the biochemical reactions that takes place happens in the cytoplasm. It is the main arena of the cellular activities. So all the cellular activities that takes place mostly in the cytoplasm. So the activities that are required to keep the cell in the living state are known to happen in the cytoplasm. That is, all the catabolic and anabolic reactions takes place in the cytoplasm itself. So this was about a brief introduction of eukaryotic and prokaryotic cell, the basis of which is the membrane-bound organelles. So membrane-bound organelles, besides the nucleus, eukaryotic cells have other membrane-bound. So this point is really important, that membrane-bound distinct structures called organelles like ER, Golgi complex, lysosome, mitochondria, and microbodies are present. While the prokaryotic cells lack such membrane-bound organelles. So in prokaryotic cells, such membrane-bound organelles are absent. That is, prokaryotic cell does not have endoplasmic reticulum, Golgi complex, lysosome, mitochondria, or microbodies like structure, which we have in the eukaryotic cell. So the ribosomes, which are the non-membrane-bound organelles, this is important. That ribosomes play an important role in protein synthesis as well. So these are non-membrane-bound cell organelles, which are found, since it is non-membrane-bound, it is found in both eukaryotes as well as in prokaryotes. And within the eukaryotic cells, ribosomes are found only in the cytoplasm. But so we know that in prokaryotic cell, we just have the cytoplasm and naked DNA. So ribosome will be present only in the cytoplasm. But in the eukaryotic cells, ribosomes are not found only in the cytoplasm, but also on the rough endoplasmic reticulum, and within which we have chloroplast and mitochondria as well. So basically, we can say that the organisms that contain a non-membrane bound organelles, like which have non-membrane bound organelles like ribosomes and non-membrane bound nucleus, are said to be prokaryotic cells, while the organism or the cells that contain the membrane-bound nuclei, that is, they have a nuclear membrane, that is, they have a distinct nuclear membrane surrounding the nuclei of the cell, are called as eukaryotic cell. However, ribosome is present in both the eukaryotic and prokaryotic cells, and based on this, since it is a non-membrane-bound organelle, it is present in both eukaryotic as well as in prokaryotic cells. And its function is, it plays an important role in the protein synthesis. So this was about the eukaryotic and prokaryotic cells. So we can start with the kingdom Monera. So we know that the kingdom Monera includes the prokaryotic organisms. So it is an taxonomic kingdom that includes all the prokaryotic organisms, primarily bacteria and archaebacteria. So all the bacteria, archaebacteria, mycoplasma, all the organisms that are prokaryotic are placed into the kingdom Monera. So the prokaryotic cells are represented by bacteria, blue-green algae, which are now known as cyanobacteria. So they were named as algae, but these are actually bacteria. So cyanobacteria, which is also known as blue-green algae, belongs to the kingdom Monera. This is really important. This might be asked because of the confusion in the naming. So blue-green algae, although the name appears to be that it belongs to the kingdom algae or it belongs to algae, but no, it is a cyanobacteria, that is, it belongs to the kingdom Monera. So the prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO. PPLO is pleuropneumonia-like organism, which is a so mycoplasma and PPLO. PPLO is itself a mycoplasma that lacks the cell wall. So we know that since Monera includes prokaryotic cell, therefore there will be no well-defined nucleus. Like nucleus will be absent, there will be no well-defined nucleus. DNA is present in the naked form. That is why they have naked DNA, as I mentioned here, that they have the naked DNA, where the DNA is lying freely into the cytoplasm without any membrane. So basically, the genetic material is basically naked, not enveloped by a nuclear membrane. So no nuclear membrane is present. No organelles like the eukaryotic ones, like as I mentioned, Golgi apparatus, endoplasmic reticulum, mitochondria are present. Prokaryotes, but the only exception is the ribosome, which is present in both eukaryotes and prokaryotes. However, prokaryotes have something unique in their form, which is the inclusions. So inclusion bodies are present in the kingdom Monera, and all prokaryotes have a cell wall. So they have a cell wall surrounding them, except surrounding the cell membrane, except in the mycoplasma. So mycoplasma, although belongs to the kingdom Monera, but it lacks the cell. So this is the basic structure of the Monera prokaryotic cell, basically, where we have a cell membrane, as you can see, out of it, and above which we have a cell wall that surrounds the cell membrane. Then inside the cell membrane, we have a semi-fluid matrix, which is known as the cytoplasm, that is filled inside the cell. And we have naked DNA, which is present, so which is the circular chromosome in the form of circular chromosome. We have cellular appendages like flagellum that helps in the movement of the, that provides the motility to the organism. Then we have certain circular, we have certain extra chromosomal material, which is the plasmid. So plasmid also confers some stability or some resistance to the bacteria. So you will learn about all these structures in detail when we will learn about when we will learn cells in details. So basically, for now, we can understand that in the prokaryotic cells, we do not have any nucleus. We just have a naked DNA that is present freely into the cytoplasm. So in addition to the genomic DNA, as I mentioned, which is the chromosomal DNA, which is present in the cytoplasm, bacteria may have small circular DNA outside the genomic DNA, and these small DNA are called as plasmids. So the plasmid DNA confers certain unique phenotypic characters to such bacteria. For example, they may provide the resistance to the antibiotics. So we know about the antibiotic resistance bacteria. So these antibiotic resistance is mainly provided by the extra chromosomal material, which is the smaller DNA, which is present outside the genomic DNA, which is called as plasmids. So these plasmids may provide resistance to certain antibiotics as they carry the antibiotic resistance gene. So basically, therefore, they provide unique ability to the bacteria for the survival, one of which is the resistance to the antibiotics. So this was about the basics of the kingdom Monera, that includes mainly the bacteria and archaebacteria. Now, the bacteria may have different shapes as well. So prokaryotes are generally smaller and they multiply more rapidly than the eukaryotic cells. They rapidly divide by the process of binary fission. So basically, they are generally smaller and vary greatly in shape and size. So they have different shape and different size as well. There are 4 basic shapes, which is coccus. So coccus is round-shaped bacteria. Bacillus, which is the rod-shaped bacteria. So we can say it is a rod-shaped bacteria. Basically, it's a rod-shaped bacteria. Then we have Vibrio. So then we have Cocobacillus, and so Cocobacillus, as the name suggests, is a combination of coccus plus bacillus. So the organisms are both rounded and rod shaped. So it is kind of oval shaped bacteria, not exactly rectangular, not exactly circular, but it's a combination of both. And then we have Spirillum, which is a spiral shaped bacteria. So a variety of shapes are conferred to the kingdom Monera. Prokaryotes have variety of shapes, and based on which they can further be classified into coccus, bacillus, vibrio, cocobacillus, and spirillum. 4 basic shapes are coccus, bacillus, vibrio, and spirillum. Cocobacillus is a combination of both coccus and bacillus. So this was about the different shapes that are present in the organisms of kingdom Monera, or we can say shapes of the prokaryotic cells. So Vibrio is comma shaped, Spirillum is spiral shaped, coccus is round shaped, Bacillus is rod shaped. So this was about the different shapes that are present in the prokaryotic cells. So we know that the kingdom Monera is divided into 2 further types of, mainly comprises of organisms of bacteria and archaebacterial group. So firstly, we will learn about the basics of the eubacteria, which is also known as the true bacteria. So basically, this is also known as the true bacteria. So basic eubacteria are the organisms with a simple cellular structure that has a cell wall, a plasma membrane, and cytoplasm. So cell structure with a cell wall, plasma membrane, and cytoplasm characterizes the bacteria. Some bacteria have flagella or villi for the movement and attachment. So as I mentioned, that the flagella and villi are the appendages that are present in the bacterial cell or in the prokaryotic cells that helps in the movement and attachment. So villi, basically villi or fimbriae attachment, and flagella helps in the movement of the bacteria. The cell wall composition. So cell wall mainly contains the peptidoglycan, which is a complex molecule providing the structural support. Based on the layer of the peptidoglycan, whether it is thick or thin, the eubacteria were further classified into 2 types, that is the gram-positive bacteria and gram-negative bacteria, about which we will learn in details. So the cell wall composition of bacteria mainly contains the peptidoglycan. So this is important, that mainly it contains the peptidoglycan, right? And this provides the structural support to the bacteria. Now the bacteria resides in the various habitats, and they are incredibly diverse and inhabit a vast area of environments. They may be present in soil, they may be present in water, they may be present in air, or they may be present inside or on the living organisms, right? Also, they can be found in extreme conditions such as hot springs, acidic environments, deep sea vents, etc. So bacteria, the habitat of bacteria is huge and diverse. It inhabits a vast area of environments, including hot springs, acidic environment, soil, water, air, and inside and outside the living organism. Reproduction, as I mentioned, prokaryotic cells reproduce fastly by the process which is known as the binary fission, where a single cell divides into 2 identical daughter cells. So we can see here, this is an example of dividing bacteria. So earlier it was a single cell, but now this constriction has happened and the single cell is divided into the 2 daughter cells, thus by the process of binary fission, each having its own cell wall and cell membrane and the naked DNA. So metabolism, again bacteria exhibit diverse metabolic pathways based on whether they are present inside the soil, water, or air. So based on their habitat also, they have adapted to different metabolism. So they may undergo photosynthesis, they may undergo aerobic respiration, anaerobic respiration, or fermentation. So the anaerobic bacteria are there which do not require oxygen for their survival, while there are certain bacteria which require oxygen for their survival. They cannot survive without oxygen. There are some bacteria that can undergo photosynthesis, therefore can have their own food. So based on the various, so This is somehow related to their diversity in the habitat also. Depending on the habitat, depending on their environmental conditions, they have adapted to different metabolic pathways. So there are many bacteria which are beneficial or harmless. Some pathogenic bacteria can cause disease in humans, animals, and plants. So the pathogenicity is there. Some pathogenic bacteria may cause diseases in humans, animals, and plants, while there are certain bacteria which are beneficial or are harmless. That is, they do not cause any harm to the human beings. So, since I mentioned that the bacteria exhibit diverse metabolic pathways, we will learn about different modes of nutrition in bacteria. So basically, bacteria exhibit diverse mode of nutrition based on. So there are 2 points based on which we can define that what is the mode of nutrition of bacteria. So the first point is how they obtain their energy, and the second point is their carbon sources. These 2 points are really important whenever we are defining the mode of nutrition in any bacteria. First thing we need to remember is how a particular group of bacteria is obtaining their energy. What is the source of their energy? And second is what is their carbon source. So based on which the primary modes of nutrition of bacteria are. The first is autotrophic. Autotrophic means we know that the bacteria which can synthesize their own food. So there are 2 types of autotrophs. First is photoautotrophs. Second is chemoautotrophs. Photoautotrophs, as the name suggests, photo means light. Chemo means chemical. So basically, phototrophs are the bacteria that use sunlight as an energy source and carbon dioxide as a carbon source for the photosynthesis. Example, cyanobacteria, which is blue-green algae. Second is chemoautotrophs. These bacteria utilize inorganic compounds as their energy source. So as the name suggests, photoautotrophs, sunlight as an energy source, chemoautotrophs, inorganic compounds as their energy source, and carbon dioxide as their carbon source. They are often found in extreme conditions, for example, hydrothermal vents and sulfur springs, where they have chemicals like sulfur to utilize inorganic compounds like sulfur compounds, where they can utilize the sulfur compounds and can use them as their energy source and can use carbon dioxide as their carbon source. So basically, autotrophs are the bacteria that are using carbon dioxide as their carbon source. But based on whether they are using sunlight as an energy source or inorganic compounds as an energy source, they are classified into photoautotrophs and chemoautotrophs. The second is the heterotrophic bacteria. So again, they are divided into 2 groups, photoautotrophs and chemoheterotrophs. So photoheterotrophs are the ones that use light as an energy source but require organic compounds as a carbon source. This is really important that they are requiring organic compounds as carbon source instead of carbon dioxide. For example, purple non-sulfur bacteria. While chemoheterotrophs are, so most of the bacteria falls into the category of chemoheterotrophs. They obtain energy and carbon from organic compounds. So their source of energy is also organic compound, while their source of carbon is also organic compounds. So pathogenic bacteria, decomposers, and also many other common bacteria belong to this group. The next mode of nutrition is mixotrophic. As the name suggests, they can switch between the autotrophic and heterotrophic modes of nutrition depending on what is the environmental condition. So basically, they have the flexibility to switch between the autotrophic mode of nutrition and heterotrophic mode of nutrition depending on what are the environmental conditions and what are the nutrients that are present in the environment. The next is the saprophytic bacteria. Saprophytes, as the name suggests, they obtain nutrients from dead and decaying organic matter. So they play a crucial role in decomposing the organic material and recycling them to the ecosystem. The other type of mode of nutrition is the parasitic. Parasitic, that require a living host on which they survive. So they require a living host organism, unlike the dead and decaying matter. So here the host is living. Here the host is dead, right? So pathogenic bacteria causing disease in plants, animals, and humans fall into this category. For example, we can say Mycobacterium tuberculosis, right? The next is a symbiotic bacteria. Symbiotic bacteria form mutually beneficial relationship with other organisms. So, for example, the nitrogen-fixing bacteria, which is present in the symbiotic association with certain plants. And here, this bacteria obtain food and nutrition from the plant while it provides the nitrogen in the fixed form to the plant, which can be utilized by the plants. The next is a lithotrophic. As the name suggests, it uses inorganic compounds. So, by knowing the name, by knowing the literal meaning of the name, so lithotrophic, something related to the rocks. Rocks are mainly composed of inorganic compounds. So lithotrophic bacteria use inorganic compounds as their source of energy. They can oxidize minerals and derive energy from the process. This group belongs to chemolithotrophs. So basically, the chemoheterotrophs includes chemolithotrophs, right? The next is oligotrophic bacteria that thrives in the environment with low environmental conditions. Low nutrient concentration, where the low nutrient is required. So they do not require very large amount of nutrition for their survival. So basically they are adapted to survive and grow in conditions where the resources are very scarce. So in scarce condition where the resources are really low, they can survive. So these are oligotrophic bacteria. So we can see that saprophytic, parasitic, symbiotic, lithotrophic, oligotrophic are mainly a type of bacteria that we can broadly put into the heterotrophic bacterial condition, right? So these are different modes of nutrition, and the most important point is they are defined on the basis of how they obtain their energy and what is their carbon source, right? So based on which they are further classified. Now I was talking about these cells. Based on whether I was talking about the bacteria, about gram-positive and gram-negative bacteria. So we can clearly see that gram-positive bacteria have outer membrane. So we can clearly see that in gram-positive bacteria, we have peptidoglycan layer, right? So this is the peptidoglycan layer, which is present in both gram-positive and gram-negative bacteria. We can see that the depth of the peptidoglycan membrane is more in the case of gram-positive bacteria, while in case of gram-negative bacteria, this peptidoglycan layer is very thin. So we have thick peptidoglycan layer in gram-positive bacteria, while we have thin peptidoglycan membrane in the gram-negative bacteria. Rest, we can see that the gram-negative bacteria has an outer membrane which is made up of lipopolysaccharides. So it is an LPS containing outer membrane they have, right? They also have lipoproteins that allows the passage of the, they have purines that allow the passage of the nutrients from the outer cell wall to the outer membrane, to the peptidoglycan, right? So this is the difference between the gram-positive and gram-negative bacteria based on the thickness of the peptidoglycan. So in this case, we have higher amount of peptidoglycan, while in this case, we have very low amount of peptidoglycan, right? So the peptidoglycan content is very low in case of gram-negative bacteria. So why they were named as gram-positive and gram-negative? So the basis of naming is the gram staining. So gram staining is one of the crucial staining techniques in microbiology, and it was first proposed by Hans Christian Gram, who first introduced it in 1882 to identify organisms that causes pneumonia. So the test involved the use of crystal violet or methylene blue as the primary color, and the ones which are gram positive will appear purple brown under the microscope, while the gram negative will appear red. Right? So this is this we can see that when we will perform the gram staining, you will learn in the higher classes. So when we will perform the gram staining, we will see that the gram negative bacteria will appear pink in color, while the gram positive bacteria will appear purple in color. So this is the gram positive bacteria, that is purple in color, right? When we visualize it after the gram staining, while this one is pink in color, right? This was about the gram-positive and gram-negative bacteria based on their cell wall composition. Their cell wall plays an important role in why the particular bacteria is retaining the particular stain, right? So peptidoglycan layer is thick in the gram-positive bacteria, therefore it is retaining the crystal violet stain and therefore is appearing the purple or violet in color, and when we visualize it under the microscope, while in case of the gram-negative bacteria, is not able to retain the crystal violet stain and therefore appears pink. After that, we have mycoplasma as well in the monorin groups. So mycoplasma are the bacteria that lacks a cell wall, and therefore, since they do not have a cell wall, they have a flexibility in shape. Also, they are among the smallest living cells. Some of them are pathogenic and cause diseases in plants and animals. Example, Mycoplasma pneumonia that causes respiratory infection in humans, and Mycoplasma gallisepticum that causes respiratory diseases in birds. So one of the major advantage of studying the Mycoplasma is their minimal genome. So their genome size is very less. Their colonization ability helps in understanding the unique characteristics that make them valuable subjects in scientific research. And therefore, this serves as model organism for fundamental cellular process. So therefore, it is very important to understand about the mycoplasma. So mycoplasma are distinct bacteria lacking the cell wall, and they are the smallest living organisms. So these are the differences between the gram-positive, gram-negative, and mycoplasma cells. So cell wall in the gram-negative is mainly 2 to 7 nanometer thick peptidoglycan, while in this we have homogeneous cell wall, which is 20 to 80 nanometer thick, mainly composed of peptidoglycan. It consists of lipopolysaccharide as well, while the mycoplasma does not have a cell wall and peptidoglycan precursors and is enclosed just by a plasma membrane. The cell shape is sphere, oval, right, curved, etc. And similarly, gram-positive bacteria may be of such shape. While mycoplasma, since it lacks cell wall, it has flexibility in shape and is pleomorphic in shape, right? Reproduction is mainly due to the binary fission, and budding or fragmentation may also happen. Metabolism is photoautotrophic, chemolithotrophic or chemoorganoheterotrophic, while in case of gram-positive bacteria, they are mainly chemoorganoheterotrophic. And similarly, mycoplasma are also chemoorganoheterotrophic. Motility is that gram-negative bacteria are also motile. So they are motile, they may be motile or non-motile, while gram-positive bacteria are mostly non-motile, right? They lack flagella, or they have peritrichous flagella if they are motile. So peritrichous flagella means flagella is present around the complete cell. Appendages, they have pili and fimbriae and stalks. While they usually lack appendages, they may have spores or hyphae, and these lack appendages. Endospores, so they have the ability to form, they cannot form endospores. Some groups can form the endospores, while the mycoplasma cannot also form the endospores. Right, so endospores are the structures that provide the extra stability in stress condition. So gram-negative bacteria do not form endospores, while the gram-positive bacteria, some groups can form the endospores, and mycoplasma cannot form the endospores. The last category is the archaebacteria. They share similarities with bacteria in terms of lacking a true nucleus. They do not have a true nucleus and membrane-bound organelles. However, the difference is their unique lipid composition. Cell wall composition is they lack peptidoglycan. Unlike bacteria, they do not have peptidoglycan layer, and their cell membrane structure is distinctive, often composed of ether-linked lipids. So as I mentioned, that they have a unique lipid composition. This is really important. Their lipid is different. The bacterial lipid is ester-linked lipids, while the archaebacterial lipid is ether-linked lipids. This ether-linked lipids provide extra stability in harsh conditions. Usually, archaebacteria are present in the harsh environmental conditions, so this provides extra stability. So, archaebacteria are known for thriving in extreme conditions such as extreme temperatures, high salinity, acidity, or alkaline conditions, right? And these can also be found in more neutral environments like soils and ocean. So this is the structure of a normal lipid, where we can see that we have an ester bond. So this is the ester bond, right? So this is the complete ester bond. While in this case, we know that we have an ether bond. So this is the ether bond, right? COC. While here we have COOC, right? So in archilipids, glycerolipids, we have ether bonds. This ether bond provide more flexibility and more resistance to the harsh environmental conditions like high temperature, salinity, pH, etc. So this is really important that the lipids of the archaebacteria contains the ether bond. So this is really important because this is a major distinction point between the archaelipids and the normal bacterial lipids, that they have ether bonds while the normal bacteria have external, right? Metabolism, again, they have a variety of metabolic pathways and can perform photosynthesis or can chemosynthesize. They contribute to nutrient cycling in ecosystem. Reproduction is similar to bacteria. They produce through binary fission, where a single cell divided into two daughter cells, identical daughter cells, and usually they are not pathogenic and are not known to cause diseases in humans. So this was about the archaebacteria. So these were the three different classes of monerals: bacteria, archaebacteria, and mycoplasma can also be considered as a separate class or maybe included in bacteria. And these were the major features of the different classes. So this was all. So this was all about the kingdom Monera, and so the kingdom Monera were divided into the three, basically mainly into two broader classes, which is bacteria and archaebacteria. The major difference between bacteria and archaebacteria is their cell wall composition, which lies in the lipid. So there is a difference in the lipid, which is of the bacterial lipid and the archaelipid. The archaelipid mainly consists of the ether bond, while the bacterial lipid mainly consists of the ester bonds. While bacteria can further be classified into gram-positive bacteria and gram-negative bacteria based on their cell wall composition. The gram-positive bacteria has a thick peptidoglycan layer, while the gram-negative bacteria has a thin peptidoglycan layer and has an outer membrane which is made up of the lipopolysaccharide and appears pink when we stain with the gram stain. They appear pink under the microscope, while the gram-positive bacteria appears purple to violet and brownish in color under the microscope. So this was about the features of the kingdom Monera. In the next class, we will learn in details about the kingdom Protista. So with this, I would like to end today's session and thank you all for attending it. Thank you.